Hello. So today we're going to talk about gastrointestinal physiology. This is part two. So we're going to spend a lot of time talking about the liver and kind of absorption and kind of get that started. If we look at, we've been talking about, or we kind of got into the small intestine and we went over the pancreas and how the pancreas kind of contributes all of these enzymes. So we're going to keep talking about that, except we're going to get into the liver and gallbladder and how they contribute. But I kind of want to point this part out, and I think it's kind of an important point of the intestine, and that is that it has a lot of surface area. So it's anatomically arranged in these folds that kind of increase surface area. So you can kind of imagine if this was just straight, then you would just have that much surface area. But with the folds, that increases the surface area, and then on those folds, it actually has villi. Okay, so this is a villus here. So that increases surface area too. So instead of just being straight across, you have all of this surface area. And then on those villi, you have microvilli, which further increase surface area. Okay, and so this is oftentimes referred to as the brush border. Okay, so the brush border is kind of this micro environment where you have enzymes that are like a lot of like lactase, a lot of specific enzymes are secreted, and then there's further breakdown of the biomolecules, and then those are absorbed, and that's where a lot of absorption takes place, is in all the way along here, but really a lot of it takes place at that brush border. Okay, so we've moved into the small intestine. We have a flush of, I don't know if that's the right word to call it, but we have secretin that was released, and then that's going to cause the release of bicarbonate from the pancreas, and CCK, cholecystokinin, which is going to release a lot of the enzymes from the pancreas. Okay, so two different cell types are releasing those two different things. Secretin and CCK are both hormones. So the thing that we're going to move into is that CCK also has an effect on the gallbladder, okay, and for the release of bile. So we're going to kind of talk around and about that, and I guess the consequences of that and all of the stories that go along with that. So the liver is one of the big things it does is it releases bile, which helps us to break down fats. Okay, so fats are kind of a problem. Fats are insoluble, and so if you take something that's insoluble, okay, so if we have a bunch of fats here, and then you have a bunch of these water molecules, well, these water molecules like to interact with each other, and they're kind of dancing around, and so they kind of will push these fat molecules all together and form just blobs, okay, for lack of a better word, okay, large drops, okay, and then the water molecules just kind of form around it. So it's the polarity of the water molecules and that kind of electrical activity, I guess, because it's polar, and it's kind of it's got a positive end and a negative end, and so they kind of interact with each other. And fats don't have any of that, so the fats end up just as a blob, and then the water is kind of dancing around it. So they're insoluble in water, so that's kind of hard to get at with enzymes. Okay, so the enzymes would have to chew around that from the outside unless we can figure out a way to turn them into little droplets. That's called emulsification, and that is what bile does. Okay, so one of the major functions of the liver is the liver makes this bile, and we're going to get into the details of how all this takes place. But the liver makes the bile. Okay, so, and that helps make those fats soluble, and the liver does a whole lot of other things that we're also going to talk about. Okay, so it's situated around the convergence of the stomach and the duodenum. So if you can kind of imagine, this is the stomach here, and then the duodenum, I think, is like in this area. Okay, so the liver sits, well, that's the gallbladder there. But the liver sits right here where the stomach empties into, that's the large intestine, but where the stomach enters into the duodenum, into the small intestine. So there's a nice, the reason I point that out, because there's a nice little access area, and that's where the gallbladder will squeeze the bile into the duodenum. Okay, that early part of the intestine. Okay, so that's where the liver is situated. It just kind of hovers around there, surrounds that area. So it does a ton of things that we'll talk about, processes and stores nutrients. You'll see a lot of statements like this, and I'm guilty of it too, because that means something, but it's also kind of vague. So we'll look at how it does it. So hepatocytes, they make enzymes that detoxify molecules absorbed with food. Okay, serves as a filter, functions in the removal of old red blood cells. That's going to take on some meaning because that's what happens with bilirubin and jaundice when the liver fails, okay, which leads to hemoglobin processing. And so this generation of bilirubin, which is yellow in color, is from these heme groups of these broken down red blood cells. All right, so bile secretion. So bile is made of a lot of things, okay, so it has a few different things in it that we'll look at. But it's made by, and again, I reiterate, it is made by the liver and it is stored in the gallbladder. So don't get those mixed up. So the liver makes bile, the gallbladder functions to store and concentrate bile that was produced by the liver. So the liver pumps it out all day long, drip, 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 and then it's stored in the gallbladder until it's needed. And then the gallbladder will actually squeeze, when it gets that CCK signal, it will actually squeeze that bile down here into the duodenum, into the early part of the duodenum. So it stores bile until so the gallbladder stores the bile until the CCK or the vagus nerve, because the vagus nerve kind of is this over, executive manager of everything that happens. But mainly it's CCK that's getting the feedback and kind of doing it as a feedback type of mechanism. So CCK, or the vagus nerve, stimulates contraction of that gallbladder. Okay, and I have here the gallbladder. When we talk about cholecysto, so chola means bile, and cyst refers to the gallbladder. So you'll see cyst used a lot, and the cyst is just a fluid-filled sac. So that's what the gallbladder is. So that's kind of the medical terminology that's used to describe those two things. So we stick them together and we say cholecystokinin. So chola means, so it's bile gallbladder. Okay, I don't know. Maybe I made that too difficult. But that's where the name comes from. So CCK was originally discovered because of its effect on the gallbladder. So it's activated in the presence of liquid, CCK is. So remember, food or chyme moves from the stomach into the duodenum, and that is going to stimulate the release of CCK from those, I think, eye cells that are those specialized cells that are in the duodenum, and then that's going to release CCK into the blood. Okay, so and then it moves from the blood. That makes it a hormone, and it moves and it activates these activities in the gallbladder. So when CCK causes contraction of the gallbladder has to kind of open up the way. So the sphincter of Oddi relaxes, and bile is released into the duodenum where the fats are present. So the sphincter of Oddi is down here. It's not named on this particular picture, but it's named in other pictures. And so it squeezes through there and it opens that sphincter, and so you can release a whole bunch of bile all at once. So the effect is that a lot of bile is released just at the right time when we eat a meal with fats. Okay. All right, so this is a flow diagram. So the duodenum, fatty acids are present, causes that release of CCK, or the secretion, into the blood as a hormone. So your plasma levels, your blood levels of CCK increase, which causes that gallbladder secretion, causes that sphincter of Oddi to open, and bile is able to flow out. So from the gallbladder, it flows into the common bile duct, and then it joins up with the pancreas and goes into the duodenum. All right, so the role and composition of bile. So hepatocytes or liver cells secrete about 8,800 to 1,000 milliliters of bile daily. So 800 to a milliliter. So about a liter of bile is made by the liver every day. And remember, it's just sort of drip, 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 dripping it out into the gallbladder. So it's mostly made of bile salts. Now, bile salts are those things that are going to help emulsify, so we're going to talk about that later. So bile salts, and then cholesterol, and cholesterol is that's when you hear about gallstones. Gallstones are made up of like extra cholesterol. Sometimes more cholesterol gets in there, and there's a lot to it that I don't think we completely understand is how exactly cholesterol is formed, or at least we didn't. Maybe some smart people are studying it, but cholesterol is kind of made from gallstones can show up if cholesterol levels are higher in the bile. Okay, and then bile pigments. Now, bile pigments are, I mean, when we talk about a pigment, we think of something that makes things colorful, but the bile pigments are like the bilirubin, urobilin, sterobilin, the things that give color, like the yellow color to urine, the brown color to feces, and the red color. And heme, really, the bilirubin is made from heme, which gives blood its red color. That's not the purpose of them, is to be pigments. They have other purposes, but they happen to impart color to the solutions in which they reside. Okay, and then phospholipids. And remember, phospholipids are those things that make up the cell membrane. So there are a lot of that in bile. And then some ions and some other stuff. Water is in there. Okay? So bile is partially excretory, which means it's waste. Okay? So it's like if something is being made in the liver and the liver just doesn't want it anymore, if it's, some kind of toxin or what it thinks is a toxin and it wants to just get rid of it, it'll just dump it out with the bile. Okay? But it also has this digestive role, these bile salts. Those are necessary for digestion, okay, for proper digestion of fats. Otherwise, the fat would just run through you and you would not have pleasant stools. Okay, so bile salts help to emulsify these fats, make them into smaller soluble droplets. And then there is this bilirubin. Okay, so we're going to talk about that before we go back and talk about the pigments. So bilirubin is one of these pigments, these bile pigments, and one of the roles of the liver is to kind of get that bilirubin in a soluble form so that it can be excreted in bile. So if that doesn't happen, then you could have excess bilirubin in the blood and in the urine, which indicates that the liver isn't working right, or could indicate that the liver isn't working right. And because it is a pigment, then it can cause that yellow color that we see with jaundice. Okay, and we'll see some pictures of that. So bilirubin metabolism. So what happens with bilirubin? Well, it's a breakdown product. We talk about bilirubin, but it's really a breakdown product of heme. And if you remember heme is what is in the 4 subunits. There are 4 protein subunits that make up hemoglobin, and the heme kind of sits in the middle of each of those 4. And inside the heme, it has the iron, and the iron sticks to the oxygen. So maybe that rings a bell, but that's what heme is. Well, when red blood cells get old and they start to die and break apart, we don't just release them into the blood. I mean, that does happen, but those have to be broken down, and the heme is broken down into ultimately into bilirubin. Okay, so hemoglobin, the heme group, is broken down into bilirubin. And if that happens in the bloodstream, like if it happens in the spleen or something like that, then that bilirubin will have to be carried, because it's not soluble. It will stick to albumin, and that's how the bilirubin is carried around the body, is on albumin. Okay, so bilirubin is derived from the heme of those recycled red blood cells. Okay, we recycle the iron, at least, and the amino acids. So insoluble bilirubin is bound to albumin in the blood. Okay, so if it's floating around, it's not soluble, so it has to be attached to the bilirubin. But in the liver, we conjugate, we as in our bodies, conjugate or stick it to something called a glucuronic acid, and that makes it soluble, so that way it can be excreted into bile. So conjugated bilirubin is secreted into the intestines with bile, and then in the gut it's further broken down into urobilin, stercobilin. That happens in the small intestine, and that may be reabsorbed, or it may just be excreted with feces. and that those further breakdown products of bilirubin, whether it's taken back up again or excreted, but that's what imparts color to the feces. So breakdown products, stercobilin gives feces its brown color, urobilin gives urine its yellow color. Okay. All right. So we kind of see this product here, or this pathway here, bilirubin. Conjugated bilirubin can be reabsorbed, come back into circulation, and then be excreted in urine, or it's just excreted in feces, which is what we see down here. Okay. Okay, so the important parts of this pathway is what I just kind of explained. It's really about unconjugated bilirubin is in the body, stuck to the protein albumin, and the conjugated is what is excreted. Okay, so jaundice, it's a yellowish discoloration of the skin, so you can kind of see it here. A lot of times we can see it better in the eye, especially in dark skin, very dark skinned people. If it's very severe, it's still kind of apparent, unless the skin is very, very dark, and it can be pretty dramatic when it's very severe. But you can see it first and most easily in the eyes. Okay, so it results from that abnormally high accumulation of bilirubin in the blood, so the bilirubin that's attached to that albumin. So it can be caused by a lot of things. We're kind of right now just sort of talking about the liver. So when the liver isn't properly taking up that bilirubin and it isn't properly conjugating it to the glucuronic acid, and it's not secreting it the way it should or excreting it through the intestines in feces, then it collects in the body and causes that yellow color. But I mean, it kind of makes sense that excessive destruction of red blood cells, so like a hemolytic anemia where red blood cells are broken down, you can kind of imagine that there would be this buildup of bilirubin in the body. Okay, so and I put these structures here just so you can kind of see heme spelled that way, or heme, and you can just kind of see that. You don't have to know all of the processes that take place, but you can kind of see this is the bilirubin structure, and this is the conjugated structure, the soluble bilirubin that's done in the liver. Okay. All right. So free unconjugated bilirubin is actually attached to the plasma albumin. So in a lab result, it would be reported as indirect bilirubin. And conjugated bilirubin is reported as direct bilirubin, and that's, of course, the conjugated form here. So the free form, stuck to albumin, just regular bilirubin, and then conjugated bilirubin is soluble and excreted, or in some cases reabsorbed. Okay, so bile salts. So we need to talk about bile salts. That's really the important part of the digestive process of digesting fats. Okay, so we kind of get into a lot of the details. We don't have to kind of to really remember all of these steps, but I think that kind of looking at these structures and seeing that this is a process could be helpful, or it could just be confusing. But bile acids, okay, so ultimately that's what we're trying to make, are bile acids. It's just a couple of steps. Okay, so bile acids are they start out their life, or bile acids, bile salts is what we ultimately want. But bile acids are initially made from cholesterol. Okay, so this is cholesterol. Okay, and this happens in the liver, and so it kind of has the primary bile acids, and then there's a secondary form, and the secondary form is done by intestinal bacteria, and then that can be sort of reabsorbed and then and go back to the liver, and where the liver can conjugate those to a glycine, so attach a glycine or a taurine to it, and that is what we end up with as our final bile salt. Okay, so it's several steps, just so you're aware. Bile acid, and it starts out as cholesterol. Okay. So I'm not too concerned about all the details, drilling into that of how it all takes place. I do have a picture on the next page that kind of explains it a little differently. But one thing about bile salts, so bile salts is what we're ultimately, like I said, looking for. So they're amphipathic, so this word amphipathic means that they have both a hydrophobic and hydrophilic portions. And now, remember we were talking earlier about how fats are insoluble, and that's because they're hydrophobic, so they don't interact with water. And so the bile salts will kind of relate to that side with their hydrophobic side, so they'll kind of interact with fats, but they also have, the bile salts also have a hydrophilic part, which will let them interact with water, okay? Fats, okay. All right, so this is the property that allows them to act as a emulsifier, and remember our goal is to break down the big particles or the big drops into smaller droplets. Okay. All right, so let's look at that secretion and circulation of the bile salts. So if we kind of look at the steps that take place, we have the primary bile salts or bile acids that are made in the liver, and then it's kind of released and then reabsorbed. It goes back through this, what we're going to talk about later is portal circulation, back to the liver, and then remember during this stage there was another enzymatic step to make them, I think, secondary, and then back to the liver, and then they're conjugated with those amino acids, glycine, taurine, and then they're released as actual bile salts. Okay, and then those bile salts are taken back up, and so it's kind of a cyclical process. Okay, so we try to reabsorb those. I'm sure some get lost in feces, but we try to reabsorb those because they're useful. Okay, they're useful and they serve a function. All right, so most bile salts are recirculated and do not exit the body as feces. Sodium bile cotransporters are responsible for that. They absorb that bile back into the portal system in the ileum, which is late in the small intestine, which is kind of handy because that way those bile salts will remain in the intestine and kind of ride along with the fat, continuing to emulsify it so enzymes can break it down. Okay. So I have another picture here that contains all those ducts we were talking about earlier. And so if we look at what's happening, so this is the liver, and I just kind of wanted to have a picture that kind of goes along with it. So the liver is making this bile, releases it into the hepatic duct, okay, here. And then the bile is stored in the gallbladder. And then when we eat a meal, it is released into, so the cystic duct, so remember cystic refers to the gallbladder. So the cystic duct and the hepatic duct, hepatic refers to the liver, form the common bile duct. And so the common bile duct moves down here, and then it joins the pancreatic duct. So the pancreatic duct is obviously coming through the pancreas, and then they pass through that sphincter of Oddi. Okay. Now, I'll mention here, because I think it's important to kind of think about, but remember I mentioned earlier that the gallbladder can form gallstones, and these gallstones can kind of move into these ducts, okay? And they can clog this area, okay? So they can clog that for an extended period of time or a short period of time. But either way, when your gallbladder starts making these gallstones, a lot of times it's got to go after a while. It has to be removed, okay? Because if it does this, then what happens is the enzymes that the pancreas is making and trying to release are blocked from being released, and they build up in the pancreas to the point that they become active, okay? Because a lot of them can self-activate, and they'll start digesting the pancreas, okay? And that's pancreatitis, and that can be a problem with, well, I mean, it's obviously a problem. You don't want your pancreas to digest itself. Okay, so you start seeing some of those enzymes in the blood and doing things that they shouldn't do. Okay, all right, but that's the pathway, and I talk about that because it's nice to understand this pathway. So when you see that relationship between the gallbladder and gallstones and pancreatitis, and it kind of makes sense if you can kind of have that image of how things are moving through there. All right, so we'll just now talk about bile salts and how they emulsify these fats. So they emulsify the fats by surrounding them and breaking them into smaller droplets, as we have said. So we can see here, if we look at this bile salt up here, we can see that it has a hydrophobic side and a hydrophilic side. All right, so this side is hydrophobic, which means that we say that it's fat soluble, and over here is hydrophilic, and so it is water soluble. And then we have phospholipids that also kind of move into that, play that role as well. So phospholipids, if they are present, which they usually are, those have a hydrophilic side and a hydrophobic side as well. Okay, so that means that they will arrange themselves in the fat molecule with their hydrophilic side also out here with the water. So the water is out here. The water's like, okay, that's fine. You're friendly. We like you, whatever. And then the fat droplets are in between. Now, the goal of this, of course, is to have access to lipases. So enzymes should be able to kind of get in there and chew those up and actually break them down into their individual lipid parts. So monoglycerides and free fatty acids, those kinds of things. But we'll talk about that in a second. Okay, I guess we're talking about it now. So pancreatic lipase and colipase then break the fats down into smaller droplets as soluble micelles. So these are going to turn into micelles. We'll have pictures of that coming up. And then lipids at the epithelial surface leave the micelles, enter the cells lining the intestine. So that's the absorption phase. And then after absorption, they're reassembled and move into the lymph system as a chylomicron. So let's kind of look at all of these. Okay, well let's look at the different lipids that we have first. Okay, and then we'll get back and we'll talk about the micelles and the chylomicrons. Okay, so we mentioned just now, we just were kind of, when we looked at our droplets, we saw fats and we saw phospholipids. Phospholipid. Didn't realize that was misspelled. So we also have cholesterol. Okay, so we have triglycerides, cholesterol. Those are all lipids. So those all kind of end up in these droplets because these things all kind of gets pushed together, and so they're broken down by different enzymes. Okay, so these are the enzymes, the names of the enzymes that break them down. So triglycerides by lingual lipase, gastric lipase, and then there are pancreatic lipases that are released too. Okay, so they'll break them down into a monoglyceride. So that's a glycerol with just one fatty acid stuck to it, and then just free fatty acids. Okay. All right, and so that's how triglycerides are broken down. Cholesterol ester. So I have a picture of that up here. All right, so that has they're esterified, so they have an ester group that makes them less reactive. So that's how they kind of tend to be moved around. So those that when they're digested, then that ester group is kind of broken off of it, and then you can absorb it as the cholesterol and then the ester, which is a fatty acid. The phospholipids, remember those kind of have these tails. Well, that's broken down into what we call lysolecithin and then just the free fatty acids. So those are even broken down as well. Okay, so that's just how this takes place, and it kind of gives you that visual that these things are really, really broken down to their basic components. So components, so there's no activity, there's no there's nothing structurally complicated about them. Okay. So absorption. So we mentioned that they are broken down, okay, and we also mentioned that they're kind of surrounded by these bile salts. So what we end up with is this thing we call a micelle, okay, that has all of these components in it. It has cholesterol, it has the monoglycerides, it has this phospholipid, this one-legged phospholipid that we call lysolecithin. Okay? It's got that in it, and it has just free fatty acids. So here, that's what's in the mice cell. Well, we have ways of absorbing those things individually. So the cholesterol is absorbed, the monoglycerides are absorbed, and then the lyso-PL, phospholipid, is absorbed, and the free fatty acids are absorbed. Now, we want to quickly put those back together. So the cholesterol then is re-esterified. Okay? The monoglycerides are turned back into triglycerides, and the lyso-PL is turned back into just phospholipids. Okay? And that's it. So the take-home message here is that we've got they're packaged up into this solubilized form. They move to the brush border, and then they sort of interact with these epithelial cells that line the intestine, and there are transporters that pull these things across as their basic molecules, okay, that we just talked about. And then inside the cell, they're put back together, and then they can be used by the body. Okay, so they're put back together, and then they're packaged. Now this is ApoB, apoprotein, so this is a protein, and this can be used as a marker or a recognition factor for other cells. But these proteins are kind of added to this structure that's got phospholipids around the outside, because that's what phospholipids do. They sort of form a membrane. So it's got this nice phospholipid membrane with these apoproteins in there. And we're not going to get into all of the specific apoproteins, but the apoproteins kind of change based on, which structure it is. And then this structure is called a chylomicron, and it's got our cholesterol, esterified cholesterol, it's got our triglycerides, and it's got our phospholipids. And it leaves, and it moves into the lymph. Okay? So all of this stuff was absorbed, broken down, reassembled, turned into this structure that can be soluble in blood. Remember, we have that solubility problem. That's why we're turning these things into, something that has water activity on the outside. These phospholipids, they've got that hydrophilic side on the outside, but is also carrying the fat. So that moves into the lymph, and then from the lymph, it moves back into circulation. Okay. So then where does it go from there is what we're going to talk about. But this is kind of a nicer, more, I think, overview, I guess, of what's going on. So it kind of has all of the components in it. So the large fat droplets from the stomach, the emulsification by the bile salts, the formation of micelles, okay, that are small, digested structures or molecules, move to this brush border area, and then those are absorbed as their basic components, and then reformed, put back together into triglycerides, cholesterol, and phospholipids. I don't see that mentioned there, but they are there. And then they form that chylomicron. Oh, and the protein. So that's the apoprotein. Okay, apoprotein. I think B is on the chylomicron. And so that's what forms that chylomicron. So triglycerides, cholesterol, phospholipids, and the apoprotein. All right. And so that is too large. It's not able to move into the capillaries, and so it moves into what we call lacteal, but it's lymph. Okay, so it's lymph, and then it goes to the vena cava, and it is dumped into circulation, and now it's able to move through the body. So the chylomicron. So I like this slide. It kind of gives the amounts and kind of helps you visualize what we're talking about. So the chylomicron is up here. Okay, so this is the raw fats coming right out of the stomach. Okay, so it has all of these components: phospholipids, triglycerides, cholesterol, and it has the apoprotein. Okay, so a small amount of apoprotein. And now what these chylomicrons do is they will move and drop off their lipids. So this is a simplification, but it's what they do, is they drop off their lipids in adipose tissues, muscle cells, places that will use the either storage in adipose tissue, or they'll be used in like muscle cells, and they can also drop them off to liver cells. Okay, and then, okay, this was part of my homework. Then the chylomicron is the lipoprotein moves off, and the chylomicron is just kind of any pieces that are left are just kind of destroyed. Okay, so that's it. So the chylomicron drops off triglycerides, okay, primarily. Okay, it drops off triglycerides because that's mostly what it's made of, to adipose tissue, and then it just kind of is destroyed. Now, in the liver, okay, so these apoproteins, the liver is able to take all of these components, the phospholipids, triglycerides, cholesterol, and these apoproteins, and reform a what we call a very low density lipoprotein. So these are produced in the liver. Okay, so they carry the cholesterol and triglycerides that are made in the liver, from the liver to adipose tissue. Okay, so this is the goal of the VLDLs that are taking these extra fats, these extra nutrients, and then putting them into this lipoprotein and moving them to other parts of the cell. So it moves them to adipose tissue where triglycerides are dropped off, and then that will form a higher cholesterol, low density lipoprotein. Okay, so let's look at our amounts here. So the chylomicron, so that's part of, we can consider that just part of digestion. Okay, and so it's just dropping off the fats. The LDLs, the low density lipoproteins, that's what it stands for, and the VLDLs, very low density. They're called low density because the fats have a lower, I guess, weight to volume ratio, and so they are less dense. So the more protein, the more proportion of protein, or the less fat and cholesterol that are in those, then the more dense they are. Okay, so the fat is less dense, so more fat means lower density, less fat, higher protein component means higher density. All right, so let's just kind of go through this, what we need to know about them, and that really is, and this is what we need to know, is that the very low density lipoproteins are very high in triglycerides. Okay, so you can see that this chylomicron, the digestive portion, was very, very high in triglycerides, but that was dropped off. But when the VLDLs are made, they've got 60%, so over half triglycerides, and they have a lower proportion of cholesterol and a fairly low proportion of protein. Okay, and then the phospholipids are surrounding them. Now, so if we look at what's going on here, triglycerides are dropped off, so the recognition of some of these apoproteins on the outside, so these will have different apoproteins, but the recognition of these apoproteins on the outside there are receptors for those, and then the triglycerides are moved into those cells as needed. And then after they lose a lot of these triglycerides, then they form a less or a more dense, so it goes from being very low density to just low density lipoproteins. There's also intermediate density lipoproteins if you want to get complicated. But then they'll form the low density lipoproteins. Now, low density lipoproteins, the thing to look at and the thing to remember is that they are higher in cholesterol. So up here we only had 14% cholesterol and 60% triglycerides. Low density lipoproteins, the triglycerides are only 10%, not much, okay? And cholesterol takes on a much, much higher proportion, okay? So this is what we are going to see are the bad cholesterol, because this is what kind of builds up in blood vessels and forms atherosclerotic plaques. Okay. All right, so that's what we're looking at here. And then the liver also, again, with different identities of apoproteins, the liver will then produce these other lipoproteins that, if the low density were their job was to drop off cholesterol, I don't know that I mentioned that, but the role or the job, we have these LDLs for a reason, is because cells need cholesterol. Okay, so the LDLs are dropping off the cholesterol. Yeah, I didn't mention that. So they're the main carriers of cholesterol to needy cells. So the cells express a receptor that says, hey, I need, cholesterol, and so the LDLs will respond to that, and they'll drop off the cholesterol. So the trouble is, and I already mentioned that, is that excess cholesterol can collect on the walls of the arteries. So their purpose is not to form atherosclerotic plaques. That is a problem. Okay, their purpose is to drop off cholesterol. Now, if the LDL's job is to drop off cholesterol and take it to cells, HDL, their job, and if we look here, okay, cholesterol is only 20%. And they are higher in protein. So that means that they are, their job is to collect cholesterol and then bring that back to the liver. So again, the lipoproteins that are kind of surrounding it, sort of making up the outer, membrane of the lipoprotein, they have different identities that allow them to kind of recognize cells that it needs to shuttle the cholesterol away. So HDLs remove excess cholesterol from cells and vascular membranes and transport it back to the liver, and then also some endocrine cells. We'll take them there because so they also have cholesterol because anyway. So endocrine cells are producing hormones, and they also need cholesterol to produce hormones. So we're not going to spend too much time on that, but that's why it's taking them from endocrine cells, because they also have cholesterol. But their job, and that's what we need to know, is to remove the excess cholesterol. Okay, so LDLs are carrying cholesterol to cells. HDLs are removing cholesterol and bringing it back to the liver. Okay. All right, so the liver can manufacture and store cholesterol and triglycerides. So I move this to the bottom. Okay, so the liver can manufacture and store cholesterol and triglycerides. It usually cannot manufacture enough cholesterol, so these dietary sources provide the rest. And we'll see that just so you know, that's what we're talking about. That's how the cholesterol is in the liver. The cholesterol is processing, manufacturing, and actually producing cholesterol. The liver does that, and so for use in the body. Okay, and this is just these guys are just how it moves it around. Okay, so the VLDLs, triglycerides. That's what you should think. LDLs, cholesterol, taking it to the cells. HDLs, cholesterol, removing it, taking it back to the liver. Okay, chylomicrons, digestive. It's mainly taking cholesterol to storage cells and muscle. Okay. All right. So a few things about lipoproteins. Okay. So that's what the L stands for. The last L is low density. So this is a low density lipoproteins. So cholesterol can be produced by liver cells, but usually not enough. So we already mentioned that. LDLs are the main carriers of cholesterol to LDLs, so we know that. Excess cholesterol can attach on arterial walls, and really that can happen through damage of the wall. So there can be kind of damage of that internal wall, a lot of times from blood pressure issues, or anything that would cause some kind of damage. And the LDLs will kind of get stuck in there. Okay, they kind of get stuck in these areas. And then you have something called macrophages. We don't need to know all this really. Macrophages that will come down and try to eat it and clear it out. And then that doesn't work. So then we end up with like smooth muscle and stuff kind of moving over it and walling it off. It's very beautiful. Not in real life though. And then this process kind of continues to happen, and this can break, and then if it breaks, it can form a clot. So you have platelets that are moving in there. Anyway, there's just a whole bunch of stuff. You'll learn about it. And that's what causes the plaques. And it all starts with these little LDLs. Okay. All right. Which is why, let's see, and then, HDLs remove the excess cholesterol and transport it to the liver, which is the same thing we're going to see here. So LDLs are bad cholesterol. They leave the deposits as they take lipids to cells. HDLs are good cholesterol. They clean up the deposits in the blood as well as removing lipids from cells. Okay, so research shows that, so it used to be that we would just say diet is how you're going to control your cholesterol. And that can work, but it's very, very difficult for some people. So research shows that distribution of LDLs and HDLs is genetically determined as well as affected by diet and exercise. Okay, so levels of LDLs are found to be lower in diabetics, and exercise and diet can increase your HDL levels. Okay. Good to know. All right, so when things are absorbed, or in the case of the spleen, when red blood cells are broken down, whatever, we have a direct pathway straight to the liver. So remember, one of the functions of the liver was to detoxify blood. So if we kind of build from that and we say, well, if it's detoxifying blood, then maybe the stuff that is just being randomly absorbed into the body from whatever we're eating, maybe we should give that a look before we just send it into regular circulation. Okay, just maybe let the liver give it a once over and say, okay, yeah, you're good. So that's called the portal circulation. So the hepatic portal system, okay, is a specialized vasculature that delivers absorbed nutrients to the liver for processing before they enter general systemic circulation. So kind of what I just said, let's absorb it. You know, we talk about, or I don't know if you've heard about, like an apple has a lot of chemicals in it, you know, things like that. And so maybe we need to check those out first. Okay, so give them processing before they enter the general systemic circulation. Direct path from the intestines and these other regions, the pancreas, from the pancreas, and from the stomach, spleen. So it detoxifies potentially bad food. Okay, so simplification, but that's what we're talking about. It also detoxify medications that we want to take and breaks them down, and sometimes forms metabolites that are useful and does all that stuff too. But usually the liver gets first crack at it. Yeah. So blood from the digestive system carries absorbed materials to the liver through the hepatic portal vein. Okay. So the portal vein. This is this guy right here. Okay, and all the tributaries to the portal vein. I'm making it red, but it's a vein. All right. So let's see. We've got. This is just kind of showing the circulation of it. So splanchnic, which is just organs in the abdominal cavity. So this is kind of showing how this takes place. So movement from the stomach, from the spleen, from the pancreas, small intestine, colon. Okay. All right, so liver receives blood from these organs, and so we need to know these: the spleen, stomach, pancreas, small intestine, colon. And I did put a note here. So this is our liver getting all of that stuff from those guys, and then it circulates back to the heart, and then of course, and then back to these cells, or to these organs, to all the organs from the heart, of course. But when blood is draining from the liver, so it's obviously getting a lot of blood all day long, or at least, it's all day long. And then it's going to pass from the liver to the vein of the liver and back to the heart. So I guess I put this note here that venous blood leaving the liver returns through the vena cava to the heart. So right-sided heart failure, I think our heart is kind of flipped around here, it looks like. But right-sided heart failure can cause congestion. So if it doesn't move back into the heart, this blood will kind of back up into this area. And you can have edema, you can have fluid buildup around this area here. Okay. So around the liver. So that's one of the consequences is hepatic edema or liver edema, fluid building up around the liver. Okay. So then you can, yeah. Okay. So if we're kind of on the subject of fluid backing up, so that was backing up from the liver to the heart. Now, if we look at our portal system, so remember we have blood coming from all of these areas and going back to the liver. That's what our portal system is. Okay. So as to keep it simple right now, if something is being absorbed by the intestines, it is moving back to the portal system, the portal vein to the liver. Now, if the liver is not functioning properly, if it is breaking down, cirrhosis, a lot of good tissue is replaced just with scar tissue. Fatty liver disease can cause it. If fat builds up in and on the liver, then, or any kind of obstruction really, any kind of resistance to flow back to the liver can cause blood flow to kind of back up into the organs. I mean, it makes sense, right? That's where it's coming from. And so if the liver isn't accepting the blood the way that it's supposed to, then it will continue to back up into these organs. And it causes something called varices. So we've kind of talked about varices as being these swollen areas, okay? And not just varices, but it can also form shunts. So blood can, it doesn't have anywhere to go. It's trying to go back to the liver, and you can see kind of, these pictures aren't great, but they kind of get the point across that it's kind of building up, and you're getting these varices. And so that blood has to go somewhere, so sometimes it'll form new vessels, and it will shunt blood from the portal system just back into regular circulation. Okay. So it'll bypass the portal system and just put it back in regular circulation, which kind of defeats the whole purpose of having the portal system. But even worse, possibly, usually, is that these varices can break. So here we have. It's not very good. So here we have. I hope that didn't screw things up. But we can see esophageal varices, and we can even see down here some hemorrhoidal varices. And these can break, and they can bleed. And if it's severe, it can cause pretty severe intestinal bleeds, okay, or GI bleeds, gastrointestinal bleeds. And that can be a problem. So obstruction of blood flow into portal circulation with portal hypertension diverts blood flow to other venous channels, including the gastric and esophageal veins. So it's backing up into esophageal varices are very common with this. Varicoses and shunts develop, releasing blood toxins to general circulation and can cause that GI bleed. All right, so let's look at how the liver itself works. I think this is an important thing to know, and it's important to be able to visualize this so when we talk about all of the things the liver does, it's less confusing, in my opinion. That's been my experience. So blood, so this is arterial blood going here. So this is pumped from the heart. And then we have the portal vein blood that's coming from, I don't know which direction it's coming, doesn't matter, but it's coming from the gut, okay, from the GI system. And so all of this blood, so remember there are things that are going around in regular circulation that we want to process. It's not just things that are coming from the portal circulation. We want to process anything that's in the blood. And so, bilirubin could be floating around in the blood. So what happens is that blood from the hepatic portal vein and the artery, the hepatic artery, they mix. So they mix kind of right here. Okay? So this is just like, hey, give us your blood, we're going to look at it, we're going to filter it. And then, so it mixes in something called sinusoids. and then the blood will move in here with all of the particles that are in it. And then we have these liver cells, hepatic cells, that are kind of lying along this, and so the blood is moving past these areas, and there are channels and carriers that will recognize certain patterns of molecules and can absorb them, or it'll like just say, hey, you're bilirubin, and I'm going to remove you from, I know this is oversimplifying, but I am going to remove you and bring you into this cell because I am producing enzymes and the components to turn you into a conjugated form. Okay. I know that's simplifying, but that's how it works. And researchers doing basic research are figuring out these pathways, but we don't have to know the pathways, we just have to know that it works. All right, so toxins from drugs, or toxins that are just toxins from the environment, whatever happens. Alcohol can move past these, and they're just taken up and then they're broken down. Okay. All right, and then if they're detoxified, okay, they may be put back into circulation. Okay. Or they may just move straight across that cell into the lymph and then into the bile duct. So this is called a canal, or bile canal, or canaliculus. And so this will then drain into the bile, okay, into the bile duct. All right, so recap. Toxins from arterial blood, circulatory blood, from the portal vein will move through these sinusoids. Anything that can be processed is taken up by a lot of times specialized cells that are expressing the DNA to make the enzymes to break those things down. Okay, and then after detoxification, they'll either be moved back into the blood, or if they're solubilized or something, back into the blood where they can be excreted through the urine or used if that's the case. Like if we're making vitamins, it's like, hey, we'll take this, we'll make vitamins, and then we'll pump the vitamins back into circulation. Amino acids, we'll make amino acids, we'll pump those back into circulation. If it's a toxin, it might then be removed into the liver or into the through the kidney, or it'll be moved into the bile and then excreted that way. Okay, and that's what it does. That's how it does all these things. I think it's amazing. All right, let's see. At the back of each hepatic cell, bile is released into the bile canaliculi, and bile is carried to the bile duct and then to the gallbladder. Okay, so that's the structure, and that's how it does all of the amazing things. So nutrient interconversion, so amino acids can be formed. Okay, so you have essential amino acids, and then you have amino acids that your body can make, and they're made in the liver, through these specialized cells, and then they're put into the bloodstream. Okay, let's see, glucose into the storage form of glycogen, that happens, and then when you need glucose, then they're broken down and the liver will release them into the blood, right? Glucose converted into fats for storage. Yep, we store fat in the liver too. Lipoprotein production, those lipoproteins that are needed to carry fats around the body. Okay, the apolipoproteins are LDLs and HDLs. Activation, hydroxylation of vitamin D. Sure, let's do that too. So we have cells that can do that. Detoxification, hepatocytes remove ammonia from protein, break it down and convert it to urea. Yep, let's put urea out there so we don't have NH3 ammonia going out there causing wreaking havoc. Okay, phagocytosis of worn out, dying red and white blood cells, so that can happen. So we can break those down, break down the heme, recycle the iron, and the amino acids. That can happen in liver cells. Okay, I feel like I'm selling you a liver cell. Like, hey, this thing does everything, All right, so synthesis of many active proteins. Albumins are made there, globulins, heparin, fibrinogen, other clotting factors. Just you can buy it on Amazon. Just buy yourself a liver. You can do everything. All right, so drug metabolism in the liver. This is one function that we specifically talk about. So the liver metabolizes lipid-soluble substances that cannot be directly excreted by the kidney. So it has its own function, and it contributes to breaking down drugs or toxins. It doesn't know the difference. But phase one is to break it down. So we have these cytochrome P450s, and these cytochrome P450s, there are a number of them. I forget how many, but they may do something like decarboxylase. They just remove a carboxyl group. That's my job. That's what I do. I remove carboxyl groups, or I may remove an OH group, dehydroxylase. And so that's what these P450s do. So, they may have an effect on a lot of different medications or drugs, and that's why, oh gosh, what is it? The CYP3, I don't remember, but one of them, like it breaks down, that's why you can't have grapefruit if you're taking statins, because the cytochrome P450, it breaks down both of those, and so it's busy. It's like, no, I have to do the grapefruit, and I don't have time to break down these other. I think it's statins, but whatever. But it'll leave them in the blood longer. That's a farm thing, and I'm not thinking farm right now. Okay, so in the case of acetaminophen. So that's an example. Acetaminophen, the liver breaks it down into a toxic residue. So you start out with, perfectly fine acetaminophen, and then it is broken down into NAQ, some toxin, so something that's, not good. So this is skull and crossbone. Okay, and then that toxin is then conjugated, mixed with something else, and it's then excreted, and it's fine. Okay. So what the problem is that if you have too much of the acetaminophen, then this toxin can build up, and it can cause destruction of the liver. So that's how acetaminophen. That's kind of an example. We didn't go into the detail we could have with that, but that's just an example of how things are broken down. So inactivation of hormones, so insulin, glucagon, we release these things, and we never talk about what happens to them afterwards. So insulin is broken down, glucagon, which is what causes an increase in sugar, insulin decreases your sugar, but those are broken down in the liver, thyroxine, steroid hormones. Okay, avoided clotting. Yeah, steroid hormones, that's an important one, because those will build up if you have liver failure. Then and they kind of have a higher increased effect. Okay, so let's see. So let's, now that we have sold everybody on a liver and we're all ordering our own special liver, or we're going to take care of our livers. That's what we're going to do. We're going to have our patients take care of their livers. So now let's talk about absorption of proteins in the stomach and small intestine. So I just put a few of these in here. These were kind of in your book. So we'll look at these, and it'll feel like review, and it'll also feel like there are a lot of details that aren't super critical. So we'll try to talk about what's important, what's not important. So protein digestion in the stomach, protein, pepsin. We need to know that, and we need to know that it's released by these chief cells. So they are absorbed as amino acids and oligopeptides. Okay? So amino, let's see, is this is the small intestine. Let's see, small intestine over here. Where is my stomach? Stomach is up here, sorry. So protein digestion, proteins. So proteins broken down by pepsin into amino acids and oligopeptides, which are longer chains of proteins. So that's really too long. We don't like them that long. So in the small intestine, the oligopeptides are further broken down into dipeptides and tripeptides and amino acids, and just regular amino acids. So, so proteins are broken down into these components. Oligopeptides are also broken down in and this happens again at the brush border, not that important. What we need to know is that the stomach doesn't completely break down proteins the way that we want them to. It's not a complete process, and that we need to complete that process with all of these other trypsin, chymotrypsin, elastase, carboxypeptidase A, We don't need to know those by name. Well, probably be good to recognize trypsin, chymotrypsin. But those are how these things are further broken down into amino acids, and that's what we need to know. Dipeptides and tripeptides. That's really all we want. 1, 2, or 3 amino acids. That's it. We don't want anything functional. We don't want anything that the immune system is going to recognize and say, oh, you're foreign, and start to form antibodies against it. We don't want that to happen. Yeah, so that's one of our goals. So antibodies can form against, longer peptide chains, and we don't want that. All right. Okay. Let's see. What about sugars? Sugars in the small intestine. So again, we saw that so they have to be absorbed as monosaccharides, and we don't want disaccharides. So we break those down first before pulling them in. So digestive enzymes secreted by the small intestine break down specific disaccharides. And note that the lactase enzyme stops being transcribed in most people, making them sensitive to lactose. Okay, so, and they're carriers and transporters specific for them. Okay, so what does all that mean? Well, so we have starch, that alpha amylase, that's your salivary amylase, is broken down into these like disaccharides, even a trisaccharide, and then those have to be further broken down into more specific enzymes. So alpha amylase is breaking down starch. Okay, to break down disaccharides in trisaccharides, then you need more specific enzymes. And a lot of times what's happening here is at the brush border or epithelial cells along the edge of the intestine. Those are the enzymes. So those are being released by the intestine. Okay. So amylase, pancreatic amylase, salivary amylase are released by, well, in saliva and by the pancreas. But these specific enzymes, so lactose is broken down by the enzyme lactase into glucose and galactose. Okay, so that happens along these brush borders, along the edge of the intestine, and then those are brought in by carriers. So SGL1, SGL2, yeah, and moved in. Okay, SGL is sodium glucose transporter, and those are also in the liver, and there are drugs that block it, but that's another story. Okay, so yeah, and then it kind of moves together in this way. So we don't have to know the specific transporters. We just have to know that we're trying to break these guys down into monosaccharides before pulling them in. Okay, so another reminder, something here is that digestion and absorption of fat-soluble vitamins. So this picture isn't showing fat-soluble vitamins, but it's an interesting picture that kind of also shows us how we make our chylomicron. Our micelles and our chylomicron, so if you want to look at that, you can. But the reason I added it to fat solubles is because fat solubles are just broken down and absorbed, or they're absorbed. We want them as the vitamin, but they're fat soluble, so they don't play nice with water. They glob up. So fat-soluble vitamins A, D, E, and K are processed just like dietary lipids. They're incorporated into the micelles, transported to the membrane of the intestinal cells, taken up, absorbed, diffuse across, and then are incorporated into chylomicrons and then carried to through circulation to various cells and to the liver. All right, so from the small intestine to the colon. We haven't talked about the colon. There's really not too much to say about the colon when you compare it with what's going on in the small intestine, which has the liver involvement, pancreas involvement, so many enzymes, so much absorption. Not a lot of that happens in the large intestine. So chyme is allowed into the colon a little at a time. Okay, so we're going through that oh gosh, I've got the ileocecal valve. Ileocecal valve. Okay, so it's little at a time through the ileocecal sphincter, And so it will remain slightly contracted, and peristaltic waves hit it, and then it relaxes, opens up, lets some chyme move through into the cecum, that first bulbous early part of the colon, or the large intestine, and then it will close again. Okay? And then the cecal distension or stretching, so stretching of the cecum, causes a local reflex, and it will cause that valve to constrict again. So it's basically the valve is opening, letting some in, the cecum says got it, and then tells the valve to close. Okay, and then that will sort of move forward through the colon. Okay, so that process, by just letting a little at a time, prevents more chyme from entering the cecum, increases digestion and absorption in the small intestine. It's like saying, okay, are you sure you're ready to move this into the colon? Are you sure you've absorbed everything you need to absorb? Broken down everything you need to break down. Okay, so it slows that progress of chyme, and it also, the valve prevents backflow. Okay, so what does happen in the large intestine? So there's some mechanical digestion, so peristalsis, so we have things moving forward, we have some segmentation, and one thing that happens in the large intestine, it happens like in the small intestine too, but is this mass peristalsis. So it drives or pushes the contents of the colon forward, just kind of moves it all forward in one big lump bulk sum. Okay, and that's what that kind of moves it into the rectum, and that's where you sort of start to have that urge that you have to defecate. Okay, so that's mechanical digestion. Chemical digestion. We have intestinal flora, and so in other words, we have our gut bacteria. So many bacteria live in the intestines, so most of that is in the large intestine, in the colon. Okay, but we also have it in the stomach. We have intestinal flora, bacteria in the stomach. We have it in the small intestine. It's just that most of it will live in this in the colon. So a lot of the digestion that takes place kind of depends on certain bacteria. Okay, so fermentation of carbohydrates, production of some B vitamins, vitamin K, and then it has its byproducts, produces hydrogen, carbon dioxide, methane gases. So that's where a lot of flatulence comes from, is from these bacteria kind of processing things and helping us kind of complete the job of digestion. Okay, so feces consists of water, undigested food, which includes cellulose, or when we talk about we need a high fiber diet, so that kind of helps things move. So that means the large intestine isn't squeezing so hard and pushing so hard that it starts to form like diverticuli or bulges out of it. So that stuff kind of just moves through there. It has a lot of, and into your feces, has a lot of microorganisms, the bacteria that's moving off, sloughed off epithelial cells. It's kind of a rough place, the inside of a gut. So a lot of what we absorb, the nutrients we absorb, are actually epithelial cells that were, especially in the small intestine, that were sloughed off of the intestine itself. So it kind of loses pieces and then digests itself. Okay, so there's mucus, but no enzymes are secreted. So we don't have any enzymatic additions in that area. So most of that's going to happen by bacterial enzymes. So yeah, feces consists of water, undigested food, microorganisms, and those epithelial cells. Okay. Kind of repeat of that bullet. Okay, and this is just kind of your picture. Extends from the ileocecal junction to the anus. So the anus is down here, consists of the cecum, colon, rectum. Cecum is the early part. The colon is your large intestine, or the whole thing can be considered your large intestine, but it's divided this way. And then the rectum, and then the anal canal. So let's talk about fluid. So one of the jobs of the large intestine is to absorb fluid. So you have the chyme that moves in from the small intestine, and it's still very liquidy. Okay, so as it moves through the large intestine, there is absorption that takes place. Now, it's a major role of the colon is to absorb water, but we don't want to get confused and say that most water is absorbed in the colon. That's not true. most water is still absorbed in the small intestine. Most absorption of everything is in the small intestine. But as far as a specialty, I guess the colon does absorb a lot of water. And so there's this graph here that kind of shows what's absorbed, okay, and the amounts of it. So if you look at just absorbed fluids in general, you can see that the colon does absorb some, but it's not nearly as much as happens in the small intestine. Okay, and then a lot of water moves through the colon and is excreted as feces. But if we look at where that fluid is coming from, well, we can see that it's coming from the small intestine itself, and pancreatic juice and bile juice. So this is all just secreted from the small intestine, secreted from the bile or from the liver, because it's producing bile. The pancreas is producing fluid and pushing that into the duodenum. Gastric juices, saliva. And then we have this smaller part down here of a couple of liters of water, okay, that we drink in a day. So we've got this almost, 10 liters, 9 to 10 liters. And really only a small portion of that is diet, and most of that is just quickly reabsorbed. It's just a carrier for the food to kind of move through your intestines. Okay, so note that much of the fluid absorbed in the small intestine and colon was initially secreted by the GI system itself, and then this gives some numbers. So 15 milliliters of chyme into the cecum, 90% of the volume is reabsorbed. And so we just have a small amount of water that's in the feces. We don't have to know that number, but I think that kind of tracks and makes sense. Okay, this also gives some numbers. So we can see that if we have 1,200 milliliters of water a day, we've got 1,500 milliliters of saliva that's put in there. 2,000. So if this stuff all made it through, and it's kind of like what happens in the kidneys. If it all went through, then we would be depleted of water within a few hours. Okay, so most of it is reabsorbed. All right, and this is our absorption summary, and the last slide. So fat-soluble vitamins are absorbed like other lipids. This kind of gives you a way to, if you would like to look at all of these things as they are absorbed. This is kind of a nice little picture. Let's see, the lacteal here, or your chylomicrons that are being absorbed, and then your amino acids. And then we can see here, this kind of shows that some short chain fatty acids actually do make it into circulation, but we're not gonna, most overwhelmingly, most fat goes through the lymph system. So water and minerals are absorbed by diffusion primarily within the intestines. And that's all we have for this section. Have a good day.